Good morning, everybody. I thank each of you for attending this session. I am a PhD student from University of Paris-Descartes, and today I would like to present some preliminary results about the immunogenicity of PAMVAC vaccine candidate. Malaria during pregnancy often leads to placental infection, especially among first-time pregnant mothers who are susceptible to sequestration of infected red blood cells into the placenta. And those infected red blood cells isolated from infected placenta show a unique adhesive phenotype that do not bind to the common CD36 receptor, but rather to glycosaminoglycan conjugated sulfate A receptor of the placenta. This sequestration can have serious consequences for both mother and child, including anemia, abortion, intrauterine growth retardation, and preterm birth. VAR2-CSA, which is expressed on the surface of infected red blood cells isolated from infected placenta, has been identified as the parasite-derived protein which mediates the CSA binding of infected red blood cells. The minimal CSA binding domain is located in ID1 DBL2 domain. And during pregnancy, women can develop antibody against the infected red blood cells that can block the adhesion of these infected red blood cells, or either coat those infected red blood cells for destruction by monocytes. Therefore, the PAMVAC vaccine comprising the ID1, DBL2, ID2 domain has been expressed as a recombinant protein in Drosophila cells in a consortium led by University of Copenhagen. This vaccine has completed phase 1 clinical trial, and in 2017, 21 volunteer exposed newly gravid women have been involved in phase 1 trial in Benin. They were randomized in 3 groups of 9 who received PAMVAC adjuvanted with alidrogel, also 9 who received PAMVAC adjuvanted with GLASC adjuvant, and the third group received the placebo. They received the vaccine at 3 times 1 month interval D0, 28, and 56. They were then followed up for 7 months after the last immunization. During this follow-up, we collect the sample, peripheral blood mononuclear cells, and also the plasma from those women before the first immunization, at day 56, the day of the third immunization, and after at month 3, 6, and 9 of the follow-up. So, the current investigation aims to answer the exploratory endpoint of the clinical trial. So, in order to detect the immune effector mechanism associated with pregnancy-associated malaria, we hypothesize here that vaccination with PAMVAC vaccine candidate can induce specific IgG secreting B cells, and this antibody may coat the infected red blood cells for their destruction by monocytes. Therefore, we first quantify the specific IgG secreting B cells using ELISpot assay. So briefly, peripheral blood molecular cells were stimulated with IL-15 and CpG, kept in culture for 6 days, and then added to antigen plate, coated plate. What do we find? On the left panel are the positive control detecting the total IgG secreting B cells. And here we do not find any statistic difference between the three groups. While on the right side, we have the specific IgG secreting B cells against PAMVAC vaccine antigen. And we found that following the second immunization, there was increase in the number of those cells, especially in GLA-SC group. This response was also increased at month 6. We next investigated whether those antibodies are able to opsonize infected red blood cells using homologous strain FCR3. We opsonize the infected red blood cells with HE-inactivated plasma samples and perform phagocytosis assay using THP1 cells. The positive cells were detected by FACS. Here, we do not find any statistical difference after immunization in each of the group. However, we do observe some trends of increasing response, and this response were relatively stable over the time. We decided now to investigate the ability of those antibodies to mediate phagocytosis by targeting the DBL2 domain of the PAMVAC vaccine antigen. Therefore, we use the biotinylation and the coating of the DBL2 domain with fluorescent beads. We opsonize them with HE-inactivated plasma samples and perform also the phagocytosis using the same cells, the THP1. Here we observe a strong phagocytosis level in response to this DBL2 domain in both adjuvant group compared to the placebo not shown here. And especially, this response was significant in the GLA-SC group when we compare to the baseline at day 56 and day 84. In summary, vaccination with PAMVAC vaccine candidates induce specific IgG secreting B cells, and those antibodies increase the opsonic phagocytosis of affected red blood cells. And also, we observe a strong phagocytosis activity of the monocytes when DBL2 domains are targeted, especially with GLA-SC adjuvant. Other functional assays using heterologous strain of P. falciparum are ongoing. We are also analyzing the flow cytometry data of the different B and T cell subsets, and we also aim to determine the ex vivo T cell response by measuring the cytokine level in culture supernatant. I would like to acknowledge all the volunteers who participate in the study, all the clinical trial team from Benin, Tübingen, University of Copenhagen. And I give a big thanks to Dr. Stephen Rogerson and all his lab team for welcoming me and supporting me for the phagocytosis assay. I also thank all the postdocs, Dr. Selome Adupo, Dr. Komik Bedanle, my supervisor, Dr. Adrian Luti, and finally, the ASTMH Travel Award Selection Committee and Bill and Melinda Gates Foundation for giving me the opportunity to attend this meeting. Thanks again.
So it's time for questions. Anyone? So have you quickly measured the IgG isotype between our hydrogel and GLA-SC? What's the trend?
Yeah.
Oh? Oh, sorry. So I'm talking about IgG subclass induced by our hydrogel versus GLA-SC.
We do measure the different IgG subclasses by ELISA. I do not have the data to show you. We also measure the different IgG subclasses in response to infected red blood cells, and we found that PAMVAC vaccine induced specifically IgG1 and IgG3 response, but those responses were very increased with adjuvant GLA-SC.
Thank you, Tatiana. I'm Whitney Harrington from Seattle Children's. Do you have any data on heterologous strain challenge or response?
I don't have the data,
Yeah.
and I cannot tell more
Okay.
about that.
Thank you, Tatiana. That was very nice. I was just wondering whether you did any control experiments where you looked at a control antigen in addition to the vaccine construct. I mean, I agree that there is an upward trend, but it would be reassuring to see that you don't see a corresponding upward trend with an irrelevant construct.
Thank you. We did that for the ELISpot assay using CIGR antigen, which is not specific for pregnancy, and we do not find a great IgG secreting B cell response.
Okay, thank you.